Great. So our next session, we've just looked at FASTQs. And we're now going to go and see how we can turn this into something we can visualize in our linear genome browser. We can start to, look at it in context of other genomic features. So it's this that we're really going to turn our FASTQ into something, more applicable to our analysis, right? Something we can use and visualize. So alignment. And this is going to be a very light course on alignment. We're not going into any really big details. We're just going to give you an overview of the tool sets available for alignment, what you really absolutely need to do and consider. Okay, if people would like more details, you can raise an issue for us on our issue page here and ask us maybe to go into more details. That said, we're going to cover the really important things in this particular session. Oh, I'm in the wrong session. This session. Okay, so in the very beginning, we said FASTA is the format that we store reference genomes in. Okay, so we saw that we have here as an example the GRCH38. This is the latest human genome build, and we're going to have more conversations about genomes and genome builds and etc. Later when we have our PacBio talk by the VGL group 2 weeks from now. But for what we consider a reference genome is this long stretch of contigs, typically in FASTA format, identify, align, lots of sequence. So this is what we want to map our FASTQ reads, our sequences from our fragments of DNA to, right? So between this, which we got from someone like Connie with our sequence information, how much we rely on it, how much we trust it, we should be able to take these and map them to where they match in this sequence. And then we've got our aligned data, right? Just need to know the position and chromosome. So what we want to do is turn this with a combination of this into a SAM file. Actually, we want a BAM file. We're going to see SAM files for the first time today rather than just BAMs. But we know from when we looked at our genomic file formats, a SAM file contains our read header, right? So the same information we saw here. This sequence is now here. Is that the same sequence? It is as well, isn't it? Oh, no, not quite. And then the quality line is here. But really importantly, once we've done the alignment, we know the chromosome it's on, the start position, and how it aligned to the genome, right? This is the chromosome start flags, but this is really important. It tells us it matched for 28. We had a deletion versus the reference, and we then matched for 72. So there's one base in here. No, sorry, there's one base in the reference which wasn't in here. This matched 100 in a row. Okay, so we want to get this type of file by using the combinations of FASTQ and our reference genome in FASTA format. So we need a suitable program to do this for us, right? And we call these aligners, and they're going to replace our unaligned reads, typically FASTQ format. I think all of these you're going to need to make FASTQ or zipped FASTQ format onto our reference genome, which we'll often accept as FASTA format, and we're going to get either a SAM or a BAM file. Ultimately, we're going to always want to get a BAM file, right? We know that SAM, they're great for us to read. They're like text files, but they're useless for computers. Not useless, but very slow. Aligner softwares can be broadly placed into two categories. Those which want to align to a genome, a long contiguous sequence. So they will align to this type of genome. The second type would be a Splice-aware aligner. Okay, so they're going to need not only our genome, but they're also going to need to know something about the gene models, because they're going to look for when a read is spliced, it runs from one junction, one exon to another, right? Because we have splicing in RNA, it means that exons aren't contiguous, they aren't in a long contiguous sequence across the genome, they are split up. Okay, popular genomic aligners, we're going to see this actually in IGV anyway. Popular genomic aligners include Bowtie, actually Bowtie 2 now, which is a really popular aligner. BWA, so the same person who brought us SAM files and SAM tools, he brought us BWA, which is his aligner. Subread, which is our aligner of choice for many reasons, and GMAP. And then for Splice-aware aligners, we really see a lot of movement here. We had Top Hat 2, which kind of extended Bowtie 2 for Splice-aware. We have now HiSat 2, which is probably the replacement for Top Hat and SpliceMap. And then you have Subjunc, which is an extension of Subread. Again, this is our choice of aligner. GSnap, which is the extension of GMAP. And the famous and ubiquitous star. So a lot of people will use star to do alignment. I may comment on this as we go along. Again, I may comment on my favorite aligners. You can make your own choices and we can discuss if people want. You can read your papers and make decisions on this, but most of these aligners under the right defaults will give very similar answers. Sorry, right defaults, right parameters will give almost identical answers, and trust us that we have done the comparisons. So like I said, and it's better to illustrate it here, Splice-aware aligners are really important for RNA-seq analysis. They are important really to do this. And this is because mRNAs, so here we have the exons, which are the actual coding, the parts where we will have RNA post-splicing. Okay, we have exons, and we can see they're separated by stretches of genomic sequence. We can't expect really to have reads here. We expect to have reads mapping from this exon to this exon. So we need to make our aligner aware that when you come across gaps in exons, you may expect to see a read mapping from here to here. Right? That's because once we get the RNA, the cDNA, all these introns have been spliced out, and they're no longer in the sequence which we actually put into Connie's sequencer. So we need to be able to deal with that appropriately, and we will use a Splice-aware aligner. So of all these aligners I mentioned, they're all external, self-contained software. So you can install HiSat, you can install Subread on its own, Bowtie 2, Star on its own. But many of these have, at some level, a wrapper. So like I showed you, we have rFASP, which is just a little bit of C above someone else's program, FASP, and allows us to use this in R in a nice fluid way. People have done the same to different levels for Bowtie, Bowtie 2. They're both wrapped up in QuasR as well. GSnap, GMapper, and GMapR. SpliceMap, which we're not going to use, is in QuasR. HiSat 2 is in its own HiSat 2 package, but also the authors of QuasR use that in order to use HiSat 2. Right, and then Subread, Subjunc, the one we really like, is in our Subread. So GMapR package offers convenient access to GeneMap, GeneSnap on Mac and Linux. Sadly not implemented on Windows. I need to probably check all these facts after we know bigwigs work on Windows now. But I don't believe that was working on Windows. QuasR offers interfaces to Bowtie 2, SpliceMap, and much more recently, HiSat 2 on Windows, Mac, Linux, and so provides both the genomic and Splice-aware aligners we want. I can tell you either using Bowtie 2 and SpliceMap is going to just blow up your computer. It's very memory unfriendly. HiSat 2 works well though. Our Bowtie 2, again, all wrapped up in it's all wrapping up the Bowtie 2 software, offers, I think it's right here, significant improvement in memory and CPU usage compared to Bowtie 2 wrapped in QuasR. The one we really like, I think hopefully the team like too, is our Subread, because this offers a nice wrapper to the Subread and the Subjunc package. And it recently, because we used to have to give this course, and we would have to have an aligner here which works on Windows, but now Subread works on Linux, Mac, and Windows. So we would recommend our Subread for the analysis. But I think we're going to show you two anyway. So in this session, we're going to take use again of some public data. Same cell line from here. If you want, you can go to their portal, which should be here somewhere. Okay, if not, we can carry on and you can just download directly the FASTQ you need from here. So speed up this processing. I've actually retrieved Align data. So I've taken this sample and I've taken around the actin beta gene for HG20, GRCH37, 38, sorry. So I know that these reads all mapped to this gene according to encode. And I've just taken out those reads and we're going to try and remap them back to the genome. I further down sampled these reads because it's very deep to only 10,000 out of the 200,000 reads which map to actin beta. Give you some idea of the scale, 200,000 reads on one gene. This data is going to be here in data/sampledactin.fastq.gis. So the R-Subread package offers a fast aligner for both the genomic and Splice-aware alignment. And I'm going to do my little cell on subread here, why we really like it. And I think Matt added a link here for the latest paper. But there's actually an even later paper here. But this is their title for their latest paper, claiming it's easier, faster, cheaper, and better for alignment and quantification of RNA-seq reads. Okay, so it's hard to argue with a comment like that. And I think what they probably wrote this paper to put in here is they want to compare themselves versus one of the leaders, which was STAR. Okay, so long been said STAR is the fastest, and they want to say that's not quite right. And we're actually also cheaper and easier. We don't use as much memory. A really nice feature, and I mentioned this, a really nice point, and I mentioned this a little bit in the FASTQ talk, as I said, we're going to show you how to trim the reads, but you don't need to. Okay, and that's because short read has internally a way of what it says is soft clipping. So when it thinks that we have poor quality or bits of the read which are most likely adapters, it is going to take care of this for us. So it's going to try and clean our reads as it does the alignment. And they actually, wrote a paper up on this to show you that this just makes no difference, and they have great results. They are still faster, cheaper, better, easier, even if you don't need to do this trimming. Okay, that is actually great because that's going to save us a ton of time, and it means we don't need to keep around this intermediate trimmed FASTQ. We can just let subread take care of this all for us. So this is a bioconductor package. Again, we can Google R-Subread. It's going to bring up R-Subread. And we have this line here. We can just copy and paste and put that into our browser. We're also going to need some reference data to actually align to. So we're going to install here the BS genome H sapiens UCSC HD38. So that's the latest build of the genome. And it's the same build of the genome they were using. So we can just load that library, and once we've installed it and loaded it, we know that the whole object is available to us. So this actual object, this name, is available to us, and we can start to extract information from it. So to build an index for mapping, we need our reference sequence in FASTA format. So we need to go from our internal representation of the sequence to this format. Okay, we know how to do that. We covered that in our lectures earlier on. We know we can extract a DNA string from our DNA string, from our BS genome object by doing our double square brackets and then the name of the chromosome we want to extract. Okay, we could do $chromosome 7 too, but you're going to see why I want to do this method here. Right, so that assigns that to a DNA string here. If I want to export a DNA string, some sequence to a FASTA file, I need to make it a DNA string set. There is no option. So then I'm going to do DNA string set, and I'm going to pass in a list. This is just because that's what DNA string set is going to accept. And it's going to have my chromosome 7 and my chromosome 7 DNA string here. Right, so I'm going to call it chromosome 7. That's the name. And then I'm going to give it the DNA string. Remember, we always need to give a name when we're doing DNA strings, and here's the name, because otherwise when we write this out, we will miss this line here, right? This identifier line. And later on, when we come to do the indexing, it's going to complain and say, look, all your chromosomes are the same name. They're actually just missing the name. So we need to do that, and we actually doing that as part of the creation here. Once I have my DNA string set, I can write that out to file, and I just need to call the file chromosome7.file. Right, FA file, so it knows it needs to be a FASTA file, and that gets written. So the R subread package requires we first build an index of our reference genome. In fact, most aligners do this. This is the, FASTA is great for us to read. You can index it. I haven't actually mentioned that, but you can index FASTA files and make FAI files. But for an aligner, it's got different tasks, right? Given the size of our read, which parts of the genome can we map uniquely? And if there are parts of the genome we can't map uniquely, maybe we don't need to include them in our index, right? What's the point? If they're so ambiguous, we could never map there. If there are places which are the same, once you've split them up into the sizes for your reads, maybe we could put them somewhere together, and we know that a read could belong to both of them. Really what we're going to do, what the indexer does, and every aligner does it differently, it's just going to repack that FASTA file into something which is more efficient for it to compare the sequence to. What is nice about things like our subread compared to STAR famously is super memory hungry, 30 gigs, 40 gigs just to run it on a full-size genomes. Our subread allows you to control this, and there is a sacrifice. We can actually control how much memory we want to use when building the index, and this is also going to control how much memory is consumed when we do our alignment. Okay, so when we build the index, we can limit it to 8 gigs by setting in our build index function one parameter, and we care that to be 8,000 because it's megabytes. And then we pass to this function the FASTA file we want to make an index from, and the name of the file for the index. All right, so we're just going to make an index called cur7. Okay, this is really important, and this is going to allow you. A lot of people, they leave this course and they say, I tried your RNA-seq and I keep on running out of memory. We show you here, lower your memory. 8 gigs is still quite a lot. You can go lower still. Once we have that aligned, once we have this index built, we can then use that index with, first off, the align function. And the align function is going to align reads to the genome, not in a splice-aware fashion. Okay. We need to specify our index name, right? It's in the same directory, so it's just still chromosome 7. We tell it the name of the file we want to map. We tell it the desired output format. Here it's going to be BAM, right? So we want to go straight to BAM. We don't want to have any intermediate file. And then we're going to say output file data, and we're going to give it our subread. We're not doing a splice-aware alignment, right? We're just using this align function and then sampled actin.bam. As soon as we have that aligned data, we have our BAM file. We still need to do 2 more operations on it before it is really usable by us. You might want to check here, because some of these aligners, if you are using other ones, will do this, the next 2 steps for you. But our subread isn't. Here we want to sort first by coordinate. So we're going to take all the reads in our BAM file and make sure that they're in the order of the position they map to in the chromosome. And then we're going to create, after we've sorted, this index file, this .bai, to go with our .bam. And it's this that allows us to use it in IGV. It's actually going to give us random access to the file. We can actually retrieve reads from any coordinate once we have that index in a really memory efficient, computationally efficient manner. So in order to do this sorting, we're going to take advantage of some of the SAM tools software. So again, the guy who wrote SAM format wrote SAM tools, and he also wrote BWA. This basically is a command line software, and it allows you to handle SAM and BAM files. We're not going to cover the command line version. It's a great piece of software, and if you wanted to go have a look, I encourage this. We're going to use the R SAM tools, and this is where they've taken all the functionality, all the C language from SAM tools, and they've put this inside R to allow us to get some of the functionality from SAM tools, some functionality in reading, writing, and then some of these operations like indexing, sorting. All right, so to get hold of this, we need to do BioC manager, install, and then we're just going to install the R SAM tools package. And then once we have that, we can load it, R SAM tools, and we have all that functionality available to us. So one of the SAM tools really useful functions they have is sort. So we can get access to SAM tools functions through R. We just need to use the sort BAM function. We're going to provide it the name of our BAM file, and it was going to do a sort for us. Kind of strangely, and I'm not quite sure why, the sort BAM function takes as an argument, this makes sense, the BAM file you want to sort, the path to the BAM file you want to sort. But slightly strangely, it also takes as an output, what you want to output, just the prefix of the sorted BAM file. So it doesn't want this extension. All right, so here if I do data and I give it the BAM file we've just aligned, or BAM file we have, and I tell it I want to create a sorted actin B, it's then going to create the file. It's going to return the name of the new file to me. It's going to be called sortedactinb.bam. It's just a pain. If you put .bam here, it's going to add a .bam, and you're going to end up with .bam, which is just annoying. As soon as we've done this sorting, we can then do the index. I think if you try and sort before, sorry, index before you sort, it's going to tell you off. It doesn't really want to index it like that. So if you do this, at this point, it's then going to go index the file and produce and write to file a sortedactinb.bam.by. And this is your binary alignment index. Okay, so that's really the file we wanted. We can use some of the functionality within SAM tools to quickly get an overview of our mapping rates, how our data is mapped, so we can get an idea of, okay, did we manage to map any reads, some reads, how well did they map? Okay, just using our quick BAM flag summary. I actually find this name a little deceptive. It's not that quick. You're going to remember it has to read your whole BAM file when it does this, so it's not super quick. Probably quicker than an alternative, so they can say quick BAM flag summary. But what this has done is going into your BAM file, and it's just seeing how much of your records are mapped. Okay. And we can see here the original BAM file. We mapped, obviously, all the reads were mapped. But, you know, sorry, we have all these, we have 10,000 records, and those were all mapped. But actually, of our data, only 97% mapped. And if we look at that, 252 reads just didn't map. So in our alignment, which we've just performed of reads, which we know mapped from ENCODE to that particular gene, we've got around 97%. Okay. If we look at our BAM and IGV, okay, this is the actin beta gene, and we take those reads from ENCODE that we know have mapped. We can see that they seem to map nicely across exons. So we seem to have reasonable signal. We're still missing some reads. And if we look now and we open up in IGV the junctions track, we can start to see some part of this problem, or at least something we're missing from our alignment. Okay, and that's because, like when I said back here, we're going to do the align. This isn't a splice-aware aligner. Okay, so now I look at this and I try and find junctions. It's just not able to map these junction positions. It's not built for it. That said, I mean, if you read that paper I sent, we're only missing a small percentage of genes. And actually they do have methods to map these. They are still mapped. They're just not showing the splice junction, right? But we want that splice junction because we want to use this later on. So one thing we could do is start to modify this now to use a Splice-aware aligner inside the R subread package. And they offer that through the subjunct function. Okay, so this is going to split up our reads and say part of the read maps to this exon, part maps to that exon, and then we have a junction between the two parts of the read which map to different exons. So it's the same input for it. We're just going to give subjunct. You can give it the chromosome here. We'll give it the FASTQ here, and we can tell it the output format's BAM, and give it our sampled actin. We have to do the same as before. Now we can do data R subread sampled actin.bam. We're going to get sorted actin B spliced, no .bam extension because it's going to add it for us. And then as soon as I have that, I can index this again. Now I have my BAI file I can open up in IGV. So if I then go look at this quick BAM flag summary, actually I'm still missing a ton of reads, right? Not a ton, but some of my reads still haven't mapped, which should have mapped according to the encode. So we have our 2% of reads which should have mapped. Right? Where have these gone exactly? Right? We've now managed to map our splice. So I'm kind of happy with that. We've got our splice junctions and all the known junctions for actin beta. But you know, encodes seem to map more. So I'm curious where those all went. And actually we can start to get an idea, and I've actually gone and found these 240 reads. What happens is that star is going to map these over from the actin beta gene to a gene, 2 genes down, where there happens to be some type of mapping occurring, or it believes this splice goes into here. It's also mapping over here. It's just mapping lots of reads, what we would say are probably unlikely configurations. Okay. So we could probably look a little bit more at this, like in comparisons. But in this case, what we would say is that these 200 reads, we wouldn't count them anyway, because we're not sure what gene they belong to. So they get thrown away at a later point. And that's really what that paper I point to is going to say. It's going to say, subread is doing a great job of actually accurately measuring your gene expression. Well, star is going to map this as hard as it can, but not necessarily in really sensible configurations. But you know, star has done a job of mapping it to that thing. So at the point that we do the counting, right, when we start to include reads which get accurately on the actin beta gene, then they're going to end up looking exactly the same. So the R-Bowtie library is another, Bowtie2 is another very popular set of aligners. And there are labs here who really like HiSat2, which is from kind of the same family. So you can get access to Bowtie2 again through R. You just need to do BioC Manager install R-Bowtie2, library R-Bowtie2. Now we have that available to us. As we saw with our subread, we need to build an index to align to first. And most of these aligners, unless they're using other aligners as part of the process, they're going to make you build an index for that specific aligner. Okay, but luckily we need the same input, right? They're all going to accept a FASTA file of our reference sequences to align to. So in this case, we can just supply to the Bowtie build argument our references here, chromosome 7.far. I tell it the Bowtie2 index, and I give it here the name of the file I want to create as an index. One thing which really annoys me about, really annoys is strong, one thing which is a pain about our Bowtie2 is that it doesn't want you to accept or pass in zipped files, or doesn't want to output computationally compressed SAM file to a BAM file, right? And that's actually because this is just built for working on the command line and adapted to run in R. So it's not as well adapted, I would say, as subread. who have really made sure we're going to get good input and good output. Oh, sorry, I just wanted to admit him. Okay, so that means that we're going to have to do some slightly painful things. So we're going to load a library here, rutils, and rutils contains a function which allows us to go from a gzip file to a standard unzipped file. So we can just run gunzip, give it the path to our fastq, and it's going to unzip it and make a sampled actin.fastq. Once we have that sample actin.fastq, now we can pass that into our bowtie2 function. This is going to take our bowtie2 index. It's going to take, we're going to specify the SAM output here. I'm just going to say sampled actin.sam. And then we're going to give it not our gzip fastq, but our standard fastq. So that's going to go make a SAM file. This is a big pain. SAM files are huge compared to BAM files, 10 times the size perhaps. So we need to go and convert that to a BAM file before we can do the sorting and the indexing anyway. Luckily, we have another function available to us from the RSAM tools as BAM. This is actually kind of efficient. It's not doing any of the work in R. It's again passing it to the C. It's going to create that file on the side. And then it's going to return, and we don't print it here, but the actual name of the file, which was turned to a BAM. Okay, so we just give it the SAM file, which was coming out of here. And then we've got our BAM file. And then we can do exactly what we did on the other file. Every time we get these BAMs, we could probably check to see the index order. And we saw that before. We could look at the header using our SAM tools, using our SAM tools. But in this case, we're just going to do sort BAM, give it the BAM file, what we want the prefix to be, no.bam, index this, and now we've got a file we can start to work with. You know, for me, not having outputting a SAM, outputting a fastq makes this a really unfriendly aligner to me, but each to their own. Aligners have a variety of different costs and benefit. Again, our bowtie2 is a significant improvement on bowtie, but it wasn't Splice-aware. It was only really built for genomic alignments, which means it's okay for things like ATAC-seq, ChIP-seq, whole genome sequencing, but it's not really going to work well for RNA-seq, ribo-seq, any time where we expect to see evidence of this splicing, right, these junctions. We're going to need to use a Splice-aware aligner. So QuasR is a wrapper package. It actually wraps a lot of different tools, and it wraps bowtie, splice map, and HiSat2. So bowtie, I've just said, please don't use. It's our bowtie2 is the choice if you wanted to do that. Splice map is a very old splice aligner, but HiSat2 is very cutting edge and used a lot. So we can use QuasR to get access to our HiSat2. And the author of our HiSat2 works with the people in QuasR. So they really want QuasR to be the route that you use it. Even though there is this RHiSat2 package, they want you to use QuasR. So like any packages, BioC Manager install QuasR, library QuasR. So the main function in QuasR is this QAlign function. And it accepts just 2 arguments or requires just 2 arguments. One is a samples file, and the other one is a reference genome. What is kind of cool of this compared to maybe our subread and our bowtie2 is that they've gone that extra mile, and you can actually just give them the name of the BS genome object, and it's going to go aligned to that, right? So we can be even more bioconductor. We don't need our own FASTA. We can just supply the BS genome object. I'm not sure if we do that here. Yeah, like here. And that will allow us then, it will internally go get all the sequence information. That's useful, but most of the time you're going to want to know or at least have that sequence you're aligning to external to R. And you can obviously, we could write out the BS genome object, or you can use your own FASTA file. So everything else so far, we write out the FASTA, the BS genome to FASTA, and then we index this. This will allow you to do that step internally. You don't have to write out the BS genome yourself. It's going to do it for you. So the sample table we need is time delimited file listing the paths to the FASTQs to be aligned, and the desired sample names. Okay, so we just need data sampledactin.fastq.guz. So we don't need to worry here about unzipping this file. It's going to do that internally. We can give it a sample name here, sampled actin. And I'm going to create a data frame from this, and I just need to write that to file, my data frame here, my file here. It's tab separated, so the separation is equals quote \t. I don't want row names here. I don't want any quotes. Don't put any quotes around my characters. This is really useful if you're doing tab separated and you want to write to something to read in Excel and your actual characters have tabs in them. Sorry, your values have tabs in them. If you don't do a quote, if you put quote equals false and you try and write out a tab separated file where you have columns where they have a tab in one of the values, that's going to get split up. If you wrote quote equals true, it won't. It's going to see the quotes and maintain them in a cell. So now with that, we can specify the reference genome. And in this case, I can just do my BS genome H. sapiens UCSC HG38. I just need to have that available. I need to have installed that. Otherwise, it doesn't know where to grab it from. But I don't need to load the library. I just need to load QuasR. And now I'm going to give it my sample table, what we saw here. Give it the name of the package to take the BS genome from. And it will go and align with. Similarly, if you wanted to make comparisons with our subread, if you already had your own FASTA, you're going to run QAlign, and you can just give it the FASTA. There is no step in creating an index here. It's going to go do that as part of this process. Okay. So that means the first time you run QAlign, potentially this is going to take a very long time. It's going to do the indexing, and it's going to be a little bit of a wait. The next time you come back, it's going to have that index internally, and it's going to remember the name because it's going to be called this. If you give it chromosome7.far, it's going to create a file called cura7. So it should take a little bit longer the first time. It's going to build the index. It hasn't told us to build an index. There is no separate function to do this. It's done internally. So finally, I think this is our last slide. Last thing we would want to look at is actually QAlign. We don't really want to take advantage of QAlign by its defaults. I probably should check that QAlign default is still Bowtie or Splice Map. But because they work with Charlotte Sonesson, who's the author of RHiSat2, you can just pass in the same information and then just specify aligner is RHiSat2, and it's going to go off and run this RHiSat2. And I can tell you that is just a copy of the same program you would run on the command line. This really nice RHiSat2 aligner, just built into an R package, which means it's easy for you guys to install the same time you install the R package, right? You don't need to worry about all these dependencies the tool may require. The installation of QAlign is going to take care of that for you. So we've got to the end, and we have some exercises here. We've still got a quarter of an hour. If anyone wants to go through the exercises, I will stay around for the next quarter of an hour. If there are any questions, do we have any questions? I just want to come back to the beginning here, just for a word on this. So this line here, memory equals 8,000. That is super important. Okay, working with full genome data. When we do this, and we're going to go do it in the RNA-seq, we'll do it in the ChIP-seq, the ATAC-seq. Right now, we're just doing one chromosome. It's going to take a little bit of time, but when we do the whole genome, we're talking a 20-minute wait to get this index. But you only need to make that index once, right? You will only need to run this step once. Once you have that, aligning, say, single-end reads with subread is super fast. So for people who want to go through this and try this now in the exercises, have some patience. I promise you, you will get through this. You may hear your laptop get a little loud, but setting this memory a bit lower may take some time. But once you've got that, aligning should be reasonably straightforward and handleable on most Macs. If you have issues, if you find that it's just blowing up, let me know. And there are tricks we can try and reduce memory. I'm happy to look at that with you. So for now, I'm going to end the session here. I have some exercises to run through. I will be around in the room, but if anyone has any questions or wants me to run through this, I don't think we're going to have time today, but I am happy to meet with you directly at the end of the sessions if you wanted to run through any problems. And again, we have that help page up here if you get stuck on anything. Help page. Here we go. If you get stuck on anything. Okay, so I'll see you in. I'll see you before. I'll be around to the end of the session, and then Matt put in the chat when our next session was 1:30 till 2:30. We'll show you how we're going to deal with this aligned data. Some of the things we can do with this. Okay.